Okay, this is your chapter 16 notes, part 3. So part 1 was on the structure of DNA, and you already did those. Part 2 is what we did on DNA replication in the classroom. And part 3 is going to be on proofreading and repairing of DNA. So during replication, DNA polymerases proofread each nucleotide against the template when it's added. In finding errors, so when they find errors, DNA polymerase will remove the nucleotide and resume synthesis. Sometimes mismatched bases evade DNA polymerase, in which case other enzymes will repair. Scientists found these when a defect in these was associated with a type of colon cancer. So basically, just like if you were publishing a book, you would send it to an editor to check for errors. Your cells sends your DNA that's been copied to an editor. And the editor is DNA polymerase or other enzymes that are, their whole purpose is to proofread and repair any errors in DNA. Errors can arise after replication, constantly subjecting our DNA to damage. So DNA replication is a really easy way for errors to happen because remember when I kind of talked about the amount of DNA that you have, if you actually wrote out every letter of your entire genome, you'd fill about 1,200 textbooks the size of your textbook with letters. So your genome is huge. So during replication, lots and lots of errors can happen, but errors can also arise after replication. For example, we're constantly subjecting our DNA to damage by chemicals. Some of those chemicals are known carcinogens that we know mutate our DNA, and others are still yet to be found. Radiation, X-ray, as well as UV radiation. When you go to receive an X-ray, they put a lead vest on the other parts of your body to limit your X-ray exposure. The X-ray tech will usually step behind a door or a glass window or something so that they are not exposed to the radiation all the time. Most of the time, these are corrected before they become permanent. When they become permanent, we call them mutations that perpetuate through additional replication. If not, they can cause an accumulation of damage, which can lead to cancer. So mutations are errors that persist and they get past the repair mechanisms. And then an accumulation of too much mutation can actually lead to cancer. There are other conditions in which excessive DNA damage can lead to other things as well. But cancer is a big one. Ectoderma pigmentosum is an inherited defect in a nucleotide excision repair. So one of those repair mechanisms for proofreading your DNA. You end up with skin cancer from UV light because they cannot repair the damage. And the unique thing about this disorder is you end up with it very young. Normally an accumulation of that damage happens over a longer period of time. With this particular disorder, since you don't have the repair mechanism, since your cells don't have the repair mechanisms to fix the errors, you end up with that cancer much sooner. The evolutionary significance of mutations. Mutations are permanent changes to the DNA. If they occur in your germ cells, egg or sperm, which give rise to the gametes, that's the egg or sperm. So the germ cells are those precursors to the egg and sperm that undergo meiosis. They can be passed on to the next generation. Some mutations can be beneficial, and natural selection can favor these mutations. Ultimately, this is what is responsible for new species. So here we're talking about they can be passed on. They can be harmful. For example, in the last one we talked about, the Ectoderma pigmentosa, that is an error in the DNA of the nucleotide excision repair gene, and that has persisted, and it ends up in the germ cells, the egg and the sperm, and that ends up being passed on to the offspring. Telomeres. Because DNA polymerase only adds the 3' end, there is no way to complete the 5' ends of the daughter strands, which means that repeated replication produces shorter and shorter DNA molecules. Prokaryotes have circular DNA, so that's not the case. DNA molecules have nucleotide sequence at the end called telomeres, which do not contain genes. It's a buffer that protects an organism's genes during replication. Telomerase catalyzes the lengthening of telomeres in germ cells, thus restoring their length and compensating for the shortening, not active in most somatic cells. So telomerase is not active in most somatic cells. So body cells have shorter and shorter ends to the DNA. We're constantly deleting the very end of that DNA. But because telomerase is active in the germ cells, that's not the case. You're going to restore that link to be passed on to your offspring. It has been proposed that the shortening of the telomeres plays a role in the aging process. And you have an article that is part of your homework in this unit on reading. It's called Cell Aging and the Cell Death, I think. Cancer cells often show unusually short telomeres. These cells divide so fast and frequently that DNA has been replicated many more times or more often than normal cells, so it makes sense that they would have shorter telomeres. So here is the end of your DNA, okay, strand, and the RNA is going to come in, and then you're going to remove the primers, and there's this little bit here that never actually gets copied. And so your DNA strand gets a little bit shorter each time you make a replication. And it's okay, because for the most part, that last bit is not coding for any genes. I want to show the bigger picture of it. And it shows it kind of continued on, and where the excess there is shorter and shorter daughter molecules. So here's shorter and shorter each time it replicates. DNA is packaged into chromosomes contained in the nucleus of the eukaryotic cell. DNA in bacteria is in a dense region called a nucleoid. It's not surrounded by a membrane. It's just an area inside the cytoplasm. DNA is called chromatin, or chromatin you might pronounce it, in the nucleus prior to mitosis, at which time it coils up into the chromosome. So we don't actually see chromosomes. Chromosomes don't exist. They're not visible until a cell is about to divide. And so here's a picture of what this would look like. So here's the DNA double helix. That DNA is wound around histones, and they form the histone plus the DNA forms a nucleosome. And they just continue to wrap and wrap and wrap until you end up with something that's a little bit more packed, called a chromosome. And here's another picture that kind of continues that on. All this packaging is just squished together, and so we end up with 46 chromosomes containing all of this DNA. We're going to be getting into later talking about how DNA is used to build proteins. So before we do that, I want to talk a little bit about the two nucleic acids that are found in our cells. These are both nucleic acids, DNA and RNA. Hopefully you remember a little bit of this from Bio 1. So I'm just going to introduce the differences between them because it's going to become important later when we start talking about protein synthesis. So DNA is a double-stranded, it's a double helix. It contains deoxyribose sugar, which is where it gets the same deoxyribonucleic acid. DNA can't leave the nucleus. Well, proteins are built on ribosomes, and so there's a disconnect between the nucleus where DNA is at and the ribosome where the protein is made. And so there is an intermediary here called RNA, and that is something that can leave the nucleus, travel to the ribosome, and initiate that protein synthesis. DNA contains the base thymine, which is going to pair with an A, and RNA contains uracil instead of thymine. A would then pair with U. And that is really important because these are the things that make DNA and RNA different. So DNA, though it's stuck in the nucleus, RNA is recognized by the cell as being just different enough to be able to leave the nucleus. And there's lots of different types of RNA. mRNA, tRNA are the main two we talk about. Well, we do talk about a little bit of rRNA. This just makes up part of the ribosome. Okay, but mRNA and tRNA are very actively involved in the process of building proteins. And I think that might be it. Okay, that is it for part 3 of your chapter 16 notes.